joining us on today's show on Myeloma Crowd Radio. I'm your host, Jenny Elstrom. We'd like to thank Takeda Oncology for their sponsorship of this program. Now, fall is very busy. We have 2 upcoming meetings that I'd like to tell you about. The first is this Saturday in Columbus, Ohio, where 3 myeloma experts will join us. So if you're in the area, please take a look at our homepage and register for that. The second is on Tuesday, October 25th. Now, this is an online live session with myeloma expert Dr. Rafael Fonseca of the Mayo Clinic and myself. We are joining Andrew Shore on Patient Power to talk about how patients can get their optimal outcomes by being advocates for themselves. So that is 6:30 p.m. Pacific time, and there will be a rebroadcast of that. So if you can't come in person to a session, you may want to consider watching a session like this. Now, with us today is one of the foremost myeloma experts who leads cutting-edge myeloma research efforts internationally, Dr. Andrzej Jakubowiak. So welcome, Dr. Jakubowiak. We're so happy you're here.
Good morning, and I'm so honored to be part of this effort and this broadcast, and happy to talk to you. And it's always my favorite to share my views and opinions with patients, which I understand is our audience.
Yes, we are thrilled to have you. This is an opportunity for us to understand what's happening and what's the very latest in research from experts, exactly what they're working on, but in patient-friendly language. So we really appreciate you taking the time out of your busy schedule. Let me give an introduction for you before we get started. Dr. Andrzej Jakubowiak is Professor of Medicine and Director of the Multiple Myeloma Program at the University of Chicago, as well as a member of the Executive Committee in the section of Hematology Oncology and on the Clinical Trial Review Committee at his facility. He's co-lead in the Hematological Malignancies Working Group, also at the University of Chicago. He is a member of the Multiple Myeloma Research Consortium, Project Review Committee, and Steering Committee, as well as a member of the Alliance, which is CALGB, which designs myeloma clinical trials, and the International Myeloma Working Group, which helps define myeloma standards globally. Dr. Jakubowiak reviews articles in publications such as the British Journal of Hematology, Leukemia and Lymphoma, Blood, and the Journal of Clinical Oncology. He's received many awards, including the MMRF Myeloma Accelerator Award and Myeloma Center of the Year Award in 2008 and 2010. He's the first honorary member of the Polish Myeloma Group and was recognized as one of the best teachers in hematology oncology by Medical Fellow. He's received distinguished honors from his home country of Poland in recognition of his major contribution not only to myeloma in Poland but in the world. Dr. Jakubowiak's primary research focus is the development of new drugs for the treatment of multiple myeloma. He is the lead investigator on a number of multi-site clinical trials for patients who are newly diagnosed, have relapsed, or have become refractory. And so we're thrilled that you're here. You're, I believe, one of the foremost experts, and we have a lot to talk about. But maybe we want to talk about first about newly diagnosed myeloma and then kind of work our way through different stages of the disease, because you've been working considerably to try to find out what is the optimal induction therapy for myeloma patients. So maybe we want to start with that.
And it's one of those probably most important emotionally charged moments and questions which come to every patient with this diagnosis and physician talking to them. And I have to maybe narrow it down by sharing a few points of research which has been done in a few years, giving us some guidance what we should be using in initial treatment of myeloma. The first important point is that at this stage, even if disease may create some problems, including some organ dysfunctions, disease is most sensitive to any therapy. And we have now therapies which are essentially 100% successful in reducing the disease by 50%. So this is great news for patients and for all of us. We have at least 4 or 5 very good choices, potentially equivalent, as we cannot really say with certainty, which one is better from the other. But I think I would like to maybe point what I think we all agreed right now is number one, that initial treatment with 3-drug combination has been consistently shown to produce superior outcome than 2-drug combination. That's number one. And the most successful combinations were combinations with proteasome inhibitor and immunomodulatory drug. And in the United States, the most commonly used combination and with longest track record in this regard and first to hit 100% response rate was a combination known as RVD, also sometimes called VRD, for Revlimid, Velcade, Dex, actually brand names, or generic names, lenalidomide, bortezomib, Dex. That is by no means, if I would discuss it with my patients, one of the top choices currently in newly diagnosed myeloma. My research evolved from that particular combination power, if I may, and its ability to achieve excellent responses and deep responses by making minor change together with my colleagues to the combination and replacing bortezomib, Velcade with carfilzomib or Kyprolis, and with combination initially known as CRD, now renamed as KRD for Kyprolis, we found that it is highly powerful, highly effective. We predicted that it may potentially produce deeper responses. And while parallel study comparing KRD to RVD have not been done, they are in progress or being planned. In direct comparison, seems to favor KRD, and that's something which eventually led in my center and number of centers to selecting KRD versus RVD as top choice, with all reservations that we have not enough data and there is a need for discussion about risks and benefits. Peripheral neuropathy and RVD, very common. On the other hand, concern for cardiovascular and pulmonary toxicities, which are much less common in RVD associated with KRD. But then, in the end, we have outstanding results with KRD, and the tolerability of the regimen for most patients is very good, with only mild to at most moderate toxicities. And for my patient population, and that's how I would discuss it with you as well, with exception of very frail patients, that's my top choice. I want to say that alternative choices like Cyborg-D or VTD in Europe are reasonable, but now in direct comparison, at least Cyborg-D is inferior than a construct with immunomodulatory drug and proteasome inhibitor like VDT.
Mm-hmm. And so when you're talking about induction therapy, this is either transplant eligible or someone who may not be getting a transplant, the same. And then how many cycles are you saying is typically used for induction therapy?
So in general, we divide patients into transplant candidates and non-transplant candidates, and that's how is across the world. Probably less so in the United States, which is our audience. I don't really see a dramatic difference in selecting the regimen for transplant candidates versus non-transplant candidates, and KRD and VRD would still be my top choices, with the exception of maybe frail patients. So that's very general comment. There is no question that data for transplant as part of initial strategy, which has been generated in late 90s, is still very strong. And in fact, last year brought number of important developments further supporting transplant as initial strategy. There was a presentation at last ASH in which French investigators showed that RVD with transplant produced longer remissions than RVD without transplant in only transplant candidates. And that was done with a relatively short course of RVD, with transplant 5 cycles, without transplant 8. But equivalency of exposure to treatment was comparable, and transplant group won. So in a way, it showed to many of us that in novel era, with even as powerful regimen as RVD, transplant seems to be adding additional value and achieving longer remission. There was no difference in survival between those two groups at the time when it was presented, but at least we have seen longer remissions, which I think for every patient is important.
Mm-hmm.
So how, to maybe answer your important other question very quickly, how long to treat for those who go on transplant? I typically would use 3 to 4 cycles, and that's what most of us would use. It's a balancing to achieve good remissions. We know the deeper the response before the transplant, the better ultimately outcome, and to limit toxicities from induction, which may potentially carry over into our strategy in post-transplant setting. So that's for transplant group. And in this particular French study, there was induction with 3 cycles. Most of us probably prefer 4 cycles. And depending what we use, that may potentially impact of what we do later and for how long. And I want to comment, because your question also included how long treat altogether. In transplant candidates, the field is completely in evolving stage, I would say. Most studies and centers will use induction for 4 cycles, then transplant, and then maintenance with single agent. Most popular in United States is lenalidomide, Revlimid. However, number of studies, including our KRD with transplant studies, seem to be indicating that extended treatment, even in transplant patients, including post-transplant period, is important. But I'm using this comment supported by data from non-transplant patients. There was 2 years ago at ASH an important now published effort by, again, French investigators, which showed that Revlimid Dex or lenalidomide Dex used for not short period, 18 months, and then every patient stopped treatment versus extended treatment until progression, showed no difference until 18 months, but at the time when those patients who were in the group of 18 months treatment stopped treatment, their duration of response dramatically dropped and then was much shorter compared to those who continued on RD, providing as a proof of principle, which I think is important phrase to use, that at least in the context of this regimen, extended treatment until progression is better than shorter treatment. Survival benefit was marginal only for this group, but it was emerging, and I think that could be an argument to be made to not necessarily continue that extended treatment. But on the other hand, there was such a dramatic and statistically significant difference in remission that many of us adopted extended treatment as a new principle of therapy. At this point, I summarize. Try to use as good regimen as possible, 3 drug regimens, and plan for extended treatment with or without transplant. That would be my summary of this segment of my commentary about initial treatment.
And then, so let me see if I can summarize for patients who might not be really up to date on the different things. So you're saying typically induction therapy was Revlimid, Velcade, Dex, and now you've used Kyprolis, Revlimid, Dex, and you're finding that to be more powerful. So the RVD, if you have Revlimid, Velcade, Dex, and if you have neuropathy issues, you know, that may not be something that you want to do. But if you have cardiovascular issues, heart conditions, or things like that, you may want to exercise caution using the Kyprolis, Revlimid, Dex. So you take that all into balance when you consider using your induction therapy. And then you are saying that transplant is still critical for patients, and you give 3 to 4 cycles of induction, and then either go to transplant, or if they're not eligible for transplant, and you keep that going. And then you've learned over time that maintenance therapy until progression is probably a good idea. Is that an accurate summary?
Yes. Yeah, and if maybe I overheard or incorrectly registered, those with cardiovascular concern should not use Kyprolis, and those with neuropathy should not use bortezomib, just to make sure. Yes,
yeah.
and it is something of importance to stress. I want to say why I use more powerful, in quotation marks, KRD as my preferred regimen versus RVD, which is outstanding regimen, and potentially other alternative choices, despite some risks.
Okay.
I think because of increased sensitivity of disease in initial phase of therapy, we are observing with KRD, especially with KRD and transplant, high, in fact, never really seen before, even with any other strategies, rates of elimination disease to not detectability. It is called complete response by our traditional criteria, no evidence of any measurable myeloma protein on urine and serum, and normalization of free light. That is achieved now with this treatment strategy in over 50% of patients without transplant, and based on RAS updates in over 70% of patients with transplant in KRD, which are really giving us sense that we are making breakthrough progress by using this strategy, and potentially justifying some risks related to this approach. In addition to this traditional measure of elimination of disease by naming it complete response, we have measured in most of our patients status of MRD, or minimal residual disease, using 2 methods, flow. We are now using very much close to Spanish technique with very high sensitivity for flow in our center, and as well as next gene sequencing method, which was previously done by Sequenta, now by Adaptive, and that is more sensitive based on the reports. And these patients who are treated with this strategy achieve ultimately close to 60 and more as last update, and we will present it again at ASH in a couple of months, MRD negative disease with stringent complete response and negative imaging studies. So imagine how great achievement it is as for these patients. They and I and patients who are treated by either physician can potentially open the door to hope that maybe potentially in some of us or some of these patients, disease was permanently eliminated. So I'm arguing that strategy is a step towards finding a cure to myeloma, and of course we need to wait longer and validate this data and bigger studies, but I think it is emerging as potentially heading in this direction. So to summarize why I have this comment about the KRD and the results, I think that if we are having disease which is traditionally considered non-curable, that's how it is written in all books and how we should probably still approach it, because we don't have really evidence of curability. But if we know that proportion of patients in those prior studies which have achieved those good responses have achieved permanent elimination of disease compatible with cure, if we then correlate that cure was associated with achieving complete response in a minimal residual disease, increasing proportion of patients with this outcome in the initial phase of therapy increases probability that the larger number of patients achieving this goal may potentially be achieving elimination of the disease. And we all see those patients in our follow-ups. In multiple publications, you see that these patients are truly eventually achieving such a good responses and a durable ultimate response.
Mm-hmm. So what I hear you saying is that because of the results that you're seeing using this different combination as upfront therapy and how deeply it's kicking the disease, you are very hopeful that it's the better. What you're saying is the better you do up front, the better you're going to do overall typically. Maybe you could stop for a minute and explain a little bit about minimal residual disease testing, because I've seen graphics where it's like an iceberg. And so you see the myeloma on the top of the iceberg, and that testing to date kind of brought it down to, can you get very good partial response, or are you getting complete response, which means like you're not showing an M spike and it's not showing up on your immunoelectrophoresis test. But then minimal residual disease is even deeper, and you mentioned that you can do it by flow cytometry or next generation sequencing. Can you elaborate on the importance of the deeper testing and finding, fewer myeloma cells in this minimal residual disease testing compared to someone who says, well, I have a stringent complete response or a complete response? And then one other question that I had is, when are you doing that MRD testing? Is that after induction? Is that after the transplant? When are you seeing those great results? Because those are fabulous results.
It's a very important question. I think, again, evolving story and evolving sense of understanding it as well as we can. Let me tackle it and maybe putting as a backdrop information that not that long ago in mid-2000 decade, we had in most strategies not more than 5% of patients achieving complete response. That has increased to about 30% in majority of the currently used strategies, and I mentioned the levels which we are seeing in KRD with or without transplant. That brought absolute need to trying to see whether there is any difference between one patient with CR and another patient with CR. And we know that achieving complete response or CR is not a guarantee of elimination of the disease. That the disease is possibly going to relapse even from complete response. So how to maybe detect better any residual disease and assess better what we have accomplished led to research on evaluating whatever is left by more sensitive methods, which are now becoming essentially standard of care. And these are, as you mentioned, flow-based minimal residual disease evaluation, which was developed by Spanish investigators and based on gene sequencing methodology developed here in the United States. What these methods are doing is by aspirating the bone marrow, they are able to detect abnormal clone of malignant cells in by flow technique 1 to 100,000 cells. In other words, if we aspirate bone marrow and 1 in 100,000 cells, cell is malignant, that would indicate that there is still some residual disease, even if immunofixation and protein electrophoresis does not show any product of this cell and there is no M spike. That's why it was found to be helpful, because we noted that if we apply this technique, we can pick up patients who are seemingly in complete response but still have some residual disease. The concern was that this is not good enough, and that's why efforts have been taken and are still in progress to go deeper. And at least by theory and by some data, next gene sequencing methodology seems to be have sensitivity of picking up 1 in million cells, which is positive for a malignant phenotype, and that is eventually our current threshold. But you mentioned iceberg. The iceberg slide, which has been shown by many of us, I show it in my talk.
Mm-hmm. I like that slide.
is showing in a very kind of a easy to comprehend way what we are talking about. And top of the iceberg, what we see over the water is maybe what we shave off when we achieve disease to absence of any monoclonal spike in urine and serum. But we know that if we apply immunofixation technique, then we pick up some spikes which have not been seen without that technique. So that goes under the water, first layer. And then when we go with the flow technique, we can go even lower under the water, but there is still part of the iceberg which we are not seeing, despite the flow technique being better. And then we think that when we go with gene sequencing, we can go even lower. And do we know whether we go to the bottom or not? We don't, but I think that that's the effort which we need to make. And I would maybe link it back to my earlier comment. I am among those treating myeloma and myeloma researchers who believe that we are at the doorsteps of finding the cure for some patients with myeloma. I hope for majority of myeloma patients. And I think that we need to refine and make this technique as good as possible, that we can use this technique with highest sensitivity level to eventually be able to say that there is no detectable disease. And there is a number of studies which have done this technique and are following the patients now for many years from that point on. And we may be able to say in 2 or 3 years from now that if patients achieve this level of response and meet some other characteristics, like, for example, negative CT PET along this finding with MRD negativity, sustain MRD for a certain period of time, these patients may achieve an ultimate goal compatible with curing the disease, and maybe we can advise some of these patients in the near future, which we are not ready yet, to stop extended treatment and stop maintenance therapy. So that's important effort from that perspective, from ultimate perspective, finding the cure. Mm-hmm. But MRD also has absolutely critical value prognostically. If it's applied in a systemic way, it may, based on the studies which have already been done or are in progress, help us early in the course of the treatment on clinical trial, make prediction what will be progression-free survival, all this median time to progression for average patient treatment with this strategy, based on MRD instead of waiting 5 or 6 or 7 years as it is nowadays at 1 or 2 year landmark. For that reason, in my place to answer your question, we measure MRD at the time when patient achieves CR, but also at landmark time point, at the end of induction, at the end of transplant, at the end of consolidation post-transplant, and then yearly.
So you're doing it multiple times, and because you're looking at that as a really primary indicator. So let me ask you some follow-up questions about that. And you kind of mentioned it already, but what you're saying is this could dramatically shift the way you run clinical trials, because right now in clinical trials, from me talking to different investigators and looking up clinical trials a lot for myeloma patients, it seems like right now you're looking at either progression-free survival, or how long did it take them to progress, or overall survival. And with the better drugs, that could be a long time, right? So you're trying to create these studies around these certain endpoints. And what you're saying is you might be able to use minimal residual disease testing, now that it's such a sensitive test, as maybe a different endpoint for your clinical trial to say, okay, we're going to look at these patients to see who achieved that and what time, and then that gives us an indication of how they'll progress and how long they'll live. Is that correct?
Absolutely correct, and absolutely important point to make to ourselves and to those who approve clinical trials, including FDA. It's work in progress. I know that many of us have worked on that endpoint being now considered as valid endpoint. FDA and those who have critical look at what we have achieved so far is giving us feedback at this point that we still have rather limited data on correlation between MRD and outcome. I think this data is already emerging to the level that I am very optimistic. In fact, that one of the next studies which are on the drawing board right now or being discussed with FDA will use MRD as at least a co-primary endpoint, which may need to be validated, which is understood by progression-free survival later on to validate that this was in fact what we were planning to accomplish and will be used as surrogate endpoint. I can translate it in a more kind of a easily understood concept. If at the end of 2 years, one study gives say 80% of patients stringent complete response and MRD negative status, and another study or treatment in different arm gives say 60% or 50%. That's only a response rate at this point, and we know that from both groups patients might be still relapsing, but we can connect those two values with historical data, how patients achieving that level of response have done over time, and we can plot statistically what the curve of progression-free survival will look for those two groups, and we can propose to regulators, to those who approve those studies, this is very solid data which can predict with this and this probability, and it is high probability nowadays, outcome at 3 or 4 years. We can use it now. We will, of course, continue to follow these patients to making sure that this assumption is confirmed in an ultimate outcome. That's an extremely important effort which we are making right now.
Well, that makes sense, that you could really speed things up that way. So another question. So now that minimal residual disease testing is out, and that's with a company called Adaptive. So if patients haven't had that, they can ask their doctor about getting that test. But how many patients would you guess, or percentage of patients are using this MRD test? And then another question, can you have low minimal residual disease and then never end up progressing? What have you seen now that you've been doing this MRD test for a while?
I will answer maybe in 2 steps. Number one, it depends on which center. In my center, every patient has MRD evaluation, and if they are on clinical trial, on standard of care, we are approaching patients who are not on clinical trial in very similar way. There is the test is CLIA approved. There is potentially in different institution availability of this test. I'm not going to go into technicalities. It's still not completely reimbursed and approved, and I think that there could be a potentially a reimbursement issue if hospital does not have a mechanism of agreement with Adaptive, or insurance companies will not accept it. Again, it is at the moment probably not as big of an issue in my understanding how Adaptive is handling those tests, but I think that that would be my quick summary where we are with this test. Flow-based MRD is done for all our patients without any concerns for reimbursement. And that's very important information. It's very close in sensitivity to what adaptive technique does. So I think that between those two tests, we have more information. So that's first part. The second part of the question is extremely important, and I will link it to what I had discussed earlier. So achieving MRD disease has prognostic value, can potentially predict elimination of the disease status if we find that sustainability of the MRD for a certain period of time can be linked to that outcome. So that's one part. But I want to stress to all the patients who are craving or have heard about MRD and are unable to get to MRD negative status that elimination disease to MRD negativity is statistically predicting better outcome, but for a given patient, still residual disease does not mean ultimately bad outcome. There is an increased probability of relapsing earlier with MRD positive status than without. But I have a lot of my patients whom I treated earlier in Michigan who continue to follow up with me. So they are 14, 15 years later who never achieved complete response and MRD negative status. They may have received a few lines of therapy, but their survival is outstanding and their outcome is probably as good as those patients who have stopped treatment in MRD negative status. But so that's the important distinction which you made by asking and framing this question. It is good to have MRD negative status for our studies to have, because we know that it can serve as a surrogate endpoint, as we discussed, and maybe potentially gave us guidance how to get to curing the disease. But I think that should not be ultimately a guide yet to achieving it in patients who, despite best efforts, are not in MRD negative status. Their outcome can still plausibly be quite good.
And I've heard some doctors say it might kick you back to, you still might have MRD positive, but it might be in like an MGUS-like state. Is that true? Can you address that at all? Is that?
Yeah.
How do you differentiate it?
Yeah, it's a concept which many of us have discussed exactly how you put it. This M spike 0.1 or 0.2 may be, distressing patients and doctors because it's still evidence of residual disease, especially if it's original M spike. But this may behave not like residual myeloma, but like MGUS, which we know may for years not progress. And I have, again, with my years of working with myeloma patients, quite good number of patients having that MGUS-like behavior with or without maintenance, never really moving up and staying at this 0.1, 0.2 and behaving like MGUS. That's true. So again, individual patient at this time, we don't have data to really change our approach based on MRD status. I am actually treating both groups of patients in a very similar way for extended time. It may change, but at the moment, that's how it is. And that's one part of my comment. And the second part is that not achieving MRD negative status does not necessarily mean that this patient may not fare quite well and may not even require another round of treatment for the rest of his or her life.
So you can still have MRD positive status and be hopeful that you can have good outcomes.
Absolutely.
So before we move on to relapse, what to do at relapse, because I do want to cover that just briefly. You mentioned that the two different inductions that have been compared and on those. When you look at standard risk versus high risk, do you see any difference when you look at the high risk genetic features or high risk types of myeloma? And then for high risk patients, do you change your number of cycles at all, like to shorten that up to get them to transplant faster? Those are two totally separate topics, so sorry I joined those questions together. But
It's an important topic and potentially controversial. So as everything what I say today, and it's my opinion and my commentary on what I know about this subject. High-risk disease is present in approximately 15 to 20% of patients using maybe most sensitive and most consistently recognized in this subgroup of patients disease. So what this means, until recently, a number of investigators and algorithm of treatments have proposed the following approach: that patients who have low risk or standard risk disease could be treated less aggressively, as their disease tends to behave not having quick or early relapses and staying longer in remission, and saving that particular
Yep. We're here.
And saving better drugs or better regimens for later. That has been part of the recommendations, for example, from MSMART algorithm and some others. Many at that time would reserve those more aggressive regimens like RVD and now KRD only for high-risk patients, or preferentially for patients. And that's something which I think is changing now. First, we have seen that there is indeed difference in outcome between good risk or standard risk patients and high-risk patients in the context of any therapy. I cannot really discuss results yet, but we will present KRD in newly diagnosed patients based on risk factors at this ASH meeting. But in short, all prior studies seem to be indicating that regardless how powerful regimen we will use, if you separate patients with high risk for a given regimen, their outcome would be somehow inferior to those who are good risk disease. So that evolved into next step of data, next step of approaching now patients in the current status of ART in myeloma, that we have realized that high-risk patients indeed need better regimens, because with less aggressive regimens, their outcome has been truly highly inferior. They do need better regimens for high risk for sure. So no question about that. High-risk patients, as good regimen you can have to treat them. But what we've learned, that good risk patients benefit at least equally from more aggressive regimens, and as high risk patients, the difference between those groups remains, but both high risk and low risk patients benefit from better strategy. It has been shown in relapse setting in a SPIRE trial, which was KRD versus RD. There was a superior outcome for KRD in this regimen in relapse setting, but when they did subset analysis based on risk, they noted that benefit of about 10 plus months was seen for more or less the same amount of months improvement in high risk and low risk patients, which provided as a proof of principle argument that we should be treating also good risk patients in more aggressive way, because they can benefit more. I illustrate this with the patients who have good risk and potentially are exploring what to do. If I were the patient and I would be told that with high-risk strategy, my life expectancy will be 10 years, with the more aggressive strategy, and say 7 years with less aggressive strategy based on all data, I would be interested in extending my life by 3 years. And it's probably, I'm using those numbers just to illustrate my point. I think we, in fact, probably relatively increase life expectancy more for high-risk patients for low-risk patients than for high-risk patients. That improvement is more even visible. So in short, to answer your question, I think that we still don't have the best strategy for high-risk patients, but we all agree that whatever we think is at the moment the best we should use for high-risk patients. But most of us also agreed that if there are no contraindications for more active regimens, also good risk patients should achieve that better strategy, and that's how I approach it in my center.
That's great. Now, when you were talking about minimal residual disease testing, you also had a clinical trial that I wanted to have you mention. So you're using ixazomib, lenalidomide, and dex versus lenalidomide alone. Do you want to tell us what that trial is, what the purpose of that trial is?
Yes. Just brief background. We generally treat and enroll patients when they are newly diagnosed or if they relapse. This trial is a novel trial in a way because we are enrolling into this trial patients who completed transplant. Eligibility is presence of minimal residual disease, and then patients are randomized to what is considered standard of care or most common choice, which is Revlimid alone maintenance for one group of patients with this minimal residual disease, and another group of patients receive ixazomib, Revlimid dex. Ixazomib is oral agent, convenient for maintenance therapy, therefore, similar to bortezomib, which is now approved as Ninlaro and has shown activity in both relapse and frontline setting with randomized study showing superiority of, we call in short this regimen IRD for ixazomib RD, versus RD, looking at progression-free survival with modest improvement of that benchmark. In newly diagnosed patients, the regimen has been shown also very active. It can be potentially considered in newly diagnosed patients based on two studies with two different schedules with randomized study comparing to RD in progress. So that's why we use IRD because it does produce good responses and extended responses for this trial. It's convenient because it's all oral. It's proven to be active. It's proven to be well tolerated. There was no major difference in toxicity between those two arms. So in a way, it was by proposing it to the patients who normally are craving for having convenience, very good approach, all oral and not, at least based on the data which we have, increasing risks. And what we are looking for at this study is to see whether at the end of 1 year of treatment with either IRD or R, there will be a difference in MRD negative disease. We anticipate that even our patients will have some conversions to negative status. This could be effect of Revlimid or ongoing effect of transplant. And we anticipate that IRD group will have more of this MRD negative patients at the end of 12 months. That is a pilot phase 2 randomized study, which will give us some new information whether that strategy actually is valid and is important, and it will provide us with some guidance for potentially intensifying treatment for those who have MRD negative status, MRD positive status, if we find that this could be beneficial. So right now, earlier discussions which I had with you was we really don't know what to do with these patients. We should be hopeful because many of them will do well. That study will give us, based on design of the study, information whether this search for elimination of the disease using this particular strategy will be beneficial. And we, of course, will look at progression-free survival down the road and some other endpoints.
And what I hear you saying overall throughout the show is that your goal is to kind of front load all these different treatments potentially. And that's what this trial is like after transplant. Then what else can we do to prevent relapse? And we'll talk about relapse briefly in a second, but that's kind of what I hear you saying is let's get the most effective drugs up front. Let's still keep the transplant because it's still showing to be more effective and deeper response. And then after we finish, continue with even a triple therapy instead of just maybe one. So that with the goal of preventing relapse, because the myeloma is the most sensitive at the beginning of the disease.
Right. And the argument can be made if you use all of the good drugs at the beginning, you may be left out with nothing to use. And that is a valid argument. I challenge this argument, and I use always example when I was having difficulty getting patients on thalidomide-based triplets 10 years ago, an argument was made, well, why don't we use thalidomide later, and at the moment let's start with VAD and see what happens. Patients which I treated more aggressively have been in remission for 6, 7, 8 years, and even if they relapse, time of thalidomide passed, and we have many more choices right now, and I was use thalidomide, and I think that race for finding new drug is generally helpful because we're getting new and new drugs. And we've been absolutely super fortunate in last years that we keep getting new and promising drugs. As you know, 4 drugs approved in 2015, and some of them extremely promising. We talked already about Ninlaro. We didn't talk about dilotuzumab, anti-CS1 antibody based on eloquent data. Panabinostat earlier, and I think what excites many of us probably most is anti-CD38 antibody daratumumab or Darzalex, which is a game changer in a way. So I think that we didn't talk about this in the frontline setting. I'm sure it will come to frontline setting, but in the relapse setting, we are achieving remissions like in combination with RD in Pollux trial, approaching 40 to 50 months. And I would like to give as a backdrop information that in most of the studies which have matured enough, the median time to progression in newly diagnosed patient has been between 2 and 3 years. So we are now in relapse setting achieving 4 to 5, even 6 years remissions by just having new agents. So I would say, okay, we may be not necessarily having super group of choices if disease relapses early, but for the most part, let's push the field. Let's make sure that progress is made. And if I am unfortunate to relapse 7 years later, who knows what's going to be happening in 7 years later? Let's get to 7 years and then worry about that later.
Right, that's fantastic. Well, in the last few minutes, I want to just leave it up to you. You have many open clinical trials that you could talk about. And we could also talk about what patients should do at relapse, because that's kind of what I talk to patients about that we would cover in the show. So I want to make sure you address it a little bit. When patients relapse, what do you suggest? And then which clinical trials would you like to share with us that are the most interesting for you?
So depends on when the relapse happens, and we always look at prior history, how quickly relapse happen, and how aggressive is the relapse. Is there high-risk disease or extramedullary disease? These are directing us in variety of choices. We are fortunate to have a lot of choices, but in general, we can always try original regimen at relapse if it worked and had sustained remission for extended period of time. I generally apply a rule of making 2 changes versus what has been the most recent therapy. For example, somebody progressing on Revlimid maintenance, first relapse, we have number of choices, and as you know, number of triplets have been approved. But for example, in my center, our priority regimen is for these patients especially who had already had previously proteasome inhibitor, to make potentially a change to more effective proteasome inhibitor like Kyprolis, and its combination at the minimum is dexamethasone or maybe with Pomalyst, and that's one of our studies which is evaluating activity of Kyprolis, Pomalyst, and dexamethasone. We presented it at ASCO, and it's still ongoing study, which is still to be published, but it has very powerful effect. Nowadays, with Darzalex, not yet approved for more than single agent, but with some insurers already accepting it, reasonable alternatives will be daratumumab-based combination with, for example, with RD, if there was no refractory status to Revlimid and dexamethasone, or with proteasome inhibitors, as we had data with Velcade-Dex. Other combinations are coming anytime. So I think that what will be potentially first, second, third relapse, which I will be using. And triplets in general work better than doublets in this type of setting. We have one very powerful doublet, Kyprolis-Dex, which was superior to Vel-Dex in Endeavor trial, but whenever we can, probably triplets will give us more efficacy and longer remission. That's how I eventually approach. And then every step, the disease needs retreatment. Is becoming more complicated and more sophisticated, always requiring review of prior treatment strategy. That's why I use clinical trials. And what we use here, for example, one study which I would like to bring to people's attention, which we have in multiple sites, including University of Chicago, is combination of Kyprolis, dexamethasone with selinexor, new class of agent, which is used in our study in those patients who are progressing on Kyprolis combination. And most of these patients have exhausted all possible combinations before. And what we found, and we presented it at prior meetings, and we will have oral session from updated results at this ASH, is that majority of patients progressing on Kyprolis combination will respond to this combination because selinexor was found to be able to overcome resistance to Kyprolis in preclinical and now in clinical setting. So this is an example. A number of other studies in our center include newer agents like venetoclax, new antibody with toxin from AbbVie, which we have. We are also moving toward CAR-Ts, and these are strategies which are in our disposal.
Mm-hmm. And then you also have an elotuzumab, lenalidomide, dexamethasone study for high-risk smoldering, right?
Right, and it is one of the important efforts which is led by Dr. Gabriel from Dana-Farber. We joined that effort. It is an approach which comes from two observations. Number one, which I mentioned, the earlier the disease, the more sensitive to treatment it is. And number two, eloquent study showed that RD and elotuzumab is superior than RD. And the next observation, which was the basis and rationale for this study, Spanish investigator published the results of their study in smoldering myeloma high risk, in which they showed that patients treated in this stage of disease with Revlimid Dex had superior control of the disease, but also superior survival compared to those who were observed. This was a placebo-controlled study, which we wouldn't do anymore, but that information is available. So we are building up on this Spanish information and study and kind of escalating intensity of smoldering myeloma as a target of more effective therapy. And I have to say that this is a first step in a direction which I'm hearing and seeing a lot around. There is a study being in development with, in fact, KRD, with or without transplant, in high-risk smoldering myeloma, which we likely to participate as well, which comes with this concept, okay, this is good regimen and work well in newly diagnosed myeloma. Dr. Landgren showed the data, outstanding result, in fact, in smoldering subsets of his KRD-treated patients. So good probability that we can achieve even better results with very aggressive therapy in this segment of the disease. But it has to be an important caution, which I want to make, done in the context of clinical trial, because there is a potential that many of these patients may not need therapy for years to come. And we don't want to be entering into this haphazardly without any evidence that this is in fact better strategy than wait and watch.
That sounded great for KRD and smoldering myeloma, and it's exciting to know that there's more progress being made right now in both newly diagnosed myeloma and earlier stages. I think it's really, really exciting. We talked a little bit earlier in the show about relapse, and I just wonder, from a patient perspective, how can patients be best prepared to consider their options either when they're newly diagnosed and everything is so new and different to them, or at relapse because they need to make decisions quickly? As an expert in myeloma, what is your opinion about that?
Well, I think they need to, first of all, have a physician with expertise in the field at a level which potentially gives them and anybody sense that this physician can navigate very difficult decision-making process and guide the patient. Most of oncologists are very qualified to make those decisions, but no question some advantages of working with key myeloma specialists in key centers like Mayo or Memorial or our center. These are physicians who are not only qualified but absolutely 100% focused on myeloma. So with that in mind, they may potentially be having the newest information, maybe even a very promising clinical trial with treatment which is not available as a standard of care. At the minimum, they may potentially provide good second opinion, reinforcing the plan implemented by primary oncologists, or maybe potentially providing additional guidance. That can be usually arranged with most of us very quickly. If we hear something like that, we do, as in our place, I can see patients next day within a week for sure, and others in my group can help, and that is typical for other places as well. I think that's important. It is a very difficult moment. I agree with that, and that's why I think getting this extra opinion is important. If they overdo it, on the other hand, that may potentially be creating a lot of uncertainty what to do, because unfortunately at any crossroad, even key opinion leaders may not always 100% agree, and that may be potentially more complicated. But if that comes to this point between 2 or 3, then the patient is usually very well advised. That's my experience, by listening to not only what is on the paper for any physician who is talking to them, but whether they feel that they have somebody whom they can really fully trust in their opinion and their way of presenting what needs to be done, and then go with that, either directly under care of this physician or having him or her involved in decision-making process.
Mm-hmm. And my final question, I think, and then I had one write-in question that I'd like to ask, is you talked earlier also about later relapse. You know, you said, well, it kind of depends on the type of relapse and how long it's been and things like that. So you hear about patients who relapse multiple times and then they add a different combination and they're still alive. You know, I have friends that are still alive 17, 20 years later, which is incredibly amazing and wonderful. But and then I have other friends who struggle with it and they're aggressive, their myeloma is more aggressive. But how do you differentiate as a clinician and researcher to know which strategy at which point in time? And maybe the MRD testing that you talked about earlier will give us some clues.
Well, we didn't talk much about it, but disease is heterogeneous. So there is at least good to understand the 2 major categories: good risk versus high-risk features, and if high risks are supported by course of the disease until that point, and that typically is associated with short-lasting responses and difficulty achieving MRD negative status, with even best therapies and recurrent pattern of relapses. These patients are in highest needs to providing them with best possible next option and potentially And on the highest list of the things which should be considered for this patient will be new agents, either recently approved of those which can potentially work in this high-risk setting, or new approaches using clinical trials. The patient you mentioned, 17 plus, and there's a lot of them like that, even with multiple relapses, but each after 4, 5, 6 years or something like that, their options are generally bigger and easier to make. They are usually able to respond to prior therapy, so you can make history of treatment assessment and then pick up based on those prior histories and how agents which have been used in the past worked, and what theoretically, at least based on this history, can work best, pick up the next regimen. And I have mentioned that earlier, I generally have a rule that I try to make at least one and preferentially 2 changes versus the prior therapy, and I'm trying to follow the rule that multiple agents, and in general this is 3 for the time being, work better than 2, and try to achieve as likely high rate of response and deep response as we can. Mm-hmm. There's unfortunately no guidance from the biology of sample, with few exceptions. There may be some emerging subsets of myeloma which we may treat preferentially with one or the other agents, but that's still work which we need to do as a researchers. So we very much empirically pick up new regimens. Yeah. Fortunately, with all of the added combinations which we have, you can imagine that those type of patients almost certainly haven't tried any of the 4 newly approved drugs. So if the part of the treatment is one of the agents which is recently approved, that usually will work very well for them.
That's terrific. Well, we're excited that there's so much more in the clinic. And I had a writing question, and then I'll let you summarize kind of what we talked about today. But Wendy asked, there are a lot of clinical trials that are doublets versus triplets. And like you mentioned earlier, we're pretty much convinced that triplets are a better choice. So is there a better way of running those trials to test different combinations?
I'm sure there is. It's a very complicated question. I very much appreciate the person who sent it. It is those trials are very important for us to establish new standards of care. They are designed at the time of their design when we truly do not know whether tested triplet versus doublet would work better, and that's by any IRB committee required test point which we need to pass, and the same at FDA level. So that's in the background, but there is no question that hypothesis before this trial is that a triplet will work better than doublet. And for the most part, that's what happened in the last few years, maybe with exception of 1 or 2 studies where those differences were so small or minimal that eventually ended up not really achieving any progress, or minimal, not significant clinically progress. How to better design the studies to make some of these advances. I have some ideas. I think we should be looking at typical endpoints in those studies, like progression-free survival and difference in toxicity in both arms of treatment, and ultimately overall survival. But these time points are usually distant. So as we discussed earlier, maybe putting surrogate endpoints early into the study may potentially limit the size of these studies and create the data which can be interpreted sooner with lesser cost. Mm-hmm. This is to some extent also driven by need for providing evidence of probability of drug combination for a given indication, and these are very hard and difficult discussions which we have internally before we propose the studies to FDA or other regulatory agencies in Europe, and eventually to our colleagues and to our patients. I'm realizing that I'm not giving really answer how to make this better, but I think that with some efforts not to allow certain studies which have obvious written on the wall answer would be one thing I probably would consider as an action point that may be difficult to implement, but that's something which I am not necessarily myself interested in doing. And number two, maybe limit those evaluations to providing the arm of treatment, for example, 3 versus 2, in a way that prior historical data does not provide high probability of success of one of these arms, which may potentially be really important to consider at the start. But then, if we have this type of situation, generally establishing those outcomes based on statistical prediction, how many patients we need to enroll, may be more difficult and may require putting more patients. So it's not easy subject to tackle. And I know that I'm not helping, but I think this is probably the best I can comment at this time.
Well, it's complicated, is what you're saying. Because you have a lot of factors to consider when you're testing certain specific things. Is there something that you'd like to add just in conclusion? We're so thankful that you're joining us today.
Well, again, I want to thank you, and I will thank you, those who are listening, for staying until end and listening to me and my opinions. It's a really great pleasure. I think that you could hear me throughout this interview and my comments that I am excited about where we are in myeloma. My excitement is maybe very much related to the fact that, in particularly in newly diagnosed myeloma, we made tremendous progress achieving in newly diagnosed myeloma requiring treatment, and in smoldering myeloma, which has high risks and is now treated in some clinical trials, high level of success, elimination of the disease to high rates never seen before, and eventually increasing hope that many of these patients may be cured, as some of the data emerging from these studies seems to be showing. I think this is the area where we need to make biggest efforts, as disease in this stage is easiest to achieve response and easiest to eliminate. And I think that the direction within the context of clinical trial to apply some of these best strategies, even in smoldering myeloma, is something I very much like and like to see continuing. We didn't talk much about it, but I think some of the agents which are currently in relapse disease are ready to be moved and are in fact evaluated in newly diagnosed myeloma, like antibodies, elotuzumab. We mentioned some of it earlier, and I think that some of the more promising newer agents, which are at the moment being evaluated in relapse disease, can make a difference. And then eventually, I think, which we didn't talk much about it, but it is linked to your earlier question, in the era where we know the disease is heterogeneous, we've got to figure out how to pick up, say we talk about KRD and RVD or CyBorD, one of these regimens based on biology of disease and certain characteristics, rather than by our preference or our experiences or comparing against historical data. That needs to be done, and as well as targeting in more difficult aspect, precision medicine, some of the abnormalities which we are seeing in biology of disease and in patients' genotype of malignant cells. And both of these efforts are in progress. I'm proud to be part of MMRF and MRC, and other organizations are joining, including yours, that we are tackling those issues in the context of clinical trial. And I'm sure that in the next even month and a half after ASH, we will be wiser and more educated about what to do. And every meeting now in this era brings new information and brings new hope and further improvements, which I have no doubt will continue. Thank you so much.
Yeah, well, thank you so much. And we just appreciate all you're doing for myeloma patients, all you're doing to drive research forward, and we're just thrilled. So thank you for joining us today and taking time out of your very busy schedule, and we just wish you the best.
Thank you so much, and very much appreciated and being invited and speaking to all of you. Thank you.
Okay, thank you so much.
Thank you, bye.
Thanks for listening to another episode of Myeloma Crowd Radio. Join us for future shows to learn more about the latest in myeloma research and what it means for you.
With the Lucky Land Slots, you can get lucky just about anywhere.
This is your captain speaking. We've got clear runway and the weather's fine, but we're just going to circle up here a while and get lucky. No, nothing like that. It's just these cash prizes add up quick. So I suggest you sit back, keep your tray table upright, and start getting lucky.
Play for free at LuckyLandSlots.com. Are you feeling lucky? No purchase necessary. Void where prohibited by law. 18+ terms and conditions apply. See website for details.